Hello, and welcome to Go Beyond A&P. If I asked you to point to an antigen, what would you point to? A virus, a bacterium, maybe a pollen grain? Most students answer with some type of microorganism, and that's completely understandable. After all, we're taught from an early age that the immune system protects us from bacteria, viruses, and other foreign invaders. But here's something that may surprise you. Your immune system doesn't actually recognize bacteria. It doesn't actually recognize viruses. It doesn't recognize cancer cells. In fact, it doesn't even recognize an entire human cell. Instead, it recognizes something much smaller, tiny molecular shapes. And once you understand that idea, almost every topic in immunology suddenly begins to make sense. So think about all the things the immune system has to deal with throughout your lifetime. It has to recognize harmless food proteins, dangerous bacteria, viruses, parasites, fungi, transplanted organs, cancer cells, and even your own tissues. That's an incredibly diverse list of targets. So how can one immune system recognize so many different things? The answer is that it isn't recognizing whole organisms at all. It's recognizing specific molecular structures that those organisms carry. Those structures are called antigens. So today we're going to explore what an antigen really is, why some molecules make better antigens than others, how B cells and T cells see the exact same pathogen in completely different ways, and why understanding antigens help explain vaccines, allergies, autoimmune diseases, blood transfusions, and some organ transplantation. So by the end of this lecture, I hope you realize that the immune system doesn't see the world in the way your eyes do. It sees an invisible landscape made of proteins, peptides, and molecular patterns. And once you begin seeing the world that way, immunology becomes much easier to understand. So let's begin with the most basic question of all. What exactly is an antigen? One of the biggest challenges in immunology isn't necessarily the biology, it's the vocabulary. You'll hear words like antigen, epitope, immunogen, hapten, and they often sound interchangeable. In reality, each has a very specific meaning, and understanding those relationships now will make the rest of the lecture much easier. So let's begin with the broadest term, antigen. An antigen is any molecule that can be specifically recognized by the adaptive immune system. That recognition might occur through an antibody, a B cell receptor, or a T cell receptor after the antigen has been processed and presented. The important word here is recognized. Now let's introduce another term, immunogen. An immunogen is an antigen that actually induces an immune response. In other words, every immunogen is an antigen, but not every antigen is an immunogen. So I like to compare this to rectangles and squares. Every square is a rectangle, but not every rectangle is a square. The same relationship exists here. Now let's zoom in on another level. The immune system almost never recognizes an entire antigen. Instead, it recognizes a very small region called an epitope, sometimes called an antigenic determinant. Think of the epitope as the face of the antigen, the exact molecular surface that an immune receptor binds. Finally, let's talk about one of the most interesting concepts in immunology, the hapten. A hapten is too small to trigger an immune response by itself, but if it attaches to a much larger carrier protein, the combination may suddenly become immunogenic. One of the classic examples is penicillin. Penicillin alone is usually too small to attract much attention from the immune system. But once it binds to proteins on the surface of your own cells, it creates an entirely new molecular structure. The immune system now recognizes that new structure as foreign, which helps explain why some drug allergies occur. So don't think of these areas as four unrelated definitions. Think of them as four pieces of the same puzzle. An epitope is part of an antigen. An Immunogen is a special type of antigen, and a hapten becomes important only after it joins a larger molecule. Those relationships will continue to appear throughout the rest of today's lecture. So the next time you hear about someone having a penicillin allergy, remember that the immune system often isn't reacting to the penicillin molecule by itself. It's reacting to the brand new antigen that's created when penicillin binds to one of the body's own proteins. So now that we understand the language of immunology, let's ask another question. What does an antigen actually look like? If someone asked you to picture a protein, what would you imagine? Many students picture a long chain of amino acids, and that's true, but only at the very beginning. Proteins don't stay long, straight chains. They twist, they bend, they fold, and as they fold, they create an incredibly complex three-dimensional landscape. And imagine flying over a mountain range. From a distance, it looks like one continuous structure, but as you get closer, you begin to notice ridges, valleys, cliffs, caves, and peaks. Proteins are remarkably similar. Their surface contains grooves, pockets, loops, and exposed regions that create unique molecular landscapes. Those landscapes are what the immune system actually recognizes. Now, look at the left side of the slide. This is a conformational epitope. Here, amino acids that may be far apart in the protein sequence, that primary structure, are brought together when the protein folds into its final three-dimensional conformational shape. If that protein unfolds or denatures, that particular epitope disappears because the three-dimensional structure has been lost. Now compare that to the right. This is a linear epitope. Instead of depending on the folded structure, a linear epitope is simply a continuous sequence of amino acids. Even if the protein loses its normal three-dimensional shape, that sequence may still be recognized, provided the amino acid sequence itself remains intact. This difference turns out to be incredibly important. B cells and antibodies often recognize conformational epitopes on intact proteins. T cells, however, generally recognize linear peptide fragments after proteins have been processed inside another cell. That distinction explains why B cells and T cells can respond to the exact same pathogen, yet see it in completely different ways. And that's where we're headed next. So if B cells and T cells encounter the exact same virus, why don't they recognize it the same way? The answer has everything to do with how each cell sees the world. Imagine you and I are standing in front of the exact same painting in an art museum. I take a few steps back so I can admire the entire work. I notice the colors, the composition, and the overall image, but you walk right up to the canvas with a magnifying glass. Instead of seeing the whole painting, you're studying individual brush strokes and tiny details. So who's looking at the painting correctly? The answer is both of us. We're simply looking at it from two different perspectives. And that's exactly what's happening with B cells and T cells. They're examining the same pathogen, but they're looking at it through two completely different lenses. Let's start with the B cell. A B cell recognizes an antigen in its natural form. And if a virus, a bacterium, or toxin is floating through the bloodstream or tissue, the B cell receptor can bind directly to it. It sees the intact three-dimensional molecule exactly as it exists in the body. In other words, the B cell is looking at the whole painting. T cells work differently. A T cell never sees the complete protein. Instead, another cell must first capture the protein, break it into tiny peptide fragments, and place one of those fragments into an MHC molecule for display. And only then can the T cell receptor inspect it. So the T cell isn't looking at the whole painting, it's examining a single brushstroke. Why would the immune system do this? Because B cells and T cells have different jobs. B cells are responsible for producing antibodies that bind directly to intact pathogens or toxins. And to do that, they need to recognize molecules exactly as they'll encounter them in the body. T cells, however, specialize in asking a different question. What's happening inside the cell? And to answer that question, they don't need to see the whole protein. They only need to examine representative peptide fragments that reveal what's occurring within the cell. So although B cells and T cells are responding to the same infection, they're gathering completely different information. One sees the landscape, the other examines individual clues. Neither perspective is better. Together, they give the immune system a much more complete picture of what's happening. And that's the theme you'll notice throughout immunology. The immune system almost never relies on a single source of information. Instead, it combines multiple perspectives before deciding how to respond. B cells and T cells are looking at the same threat, but each is asking a different question. So now we know that T cells can't recognize intact proteins, but that creates an obvious question. If they never see the whole molecule, where do those peptide fragments come from? We just learned that B cells can recognize intact antigens, while T cells require processed peptide fragments. That naturally leads to the next question. Where do those peptide fragments come from? The answer depends on one very important question. Where was the antigen found? The immune system has two major pathways for processing antigen, and choosing the correct pathway begins by answering a simple question. Did the protein originate inside the cell or outside the cell? Let's start with proteins produced inside the cell. So imagine a virus infects one of your cells. Once inside, that virus hijacks the cell's machinery and begins making viral proteins. Because those proteins are being produced within the cell, they're considered endogenous, meaning they originated from within. Those proteins will eventually encounter the MHC class 1 pathway. Now let's look at the other possibility. Suppose a dendritic cell encounters a bacterium floating throughout the tissue. It engulfs that bacterium and breaks it down inside specialized compartments. Those proteins originated outside the cell, so they're considered exogenous antigens, and those proteins enter the MHC class 2 pathway. At first glance, they may seem like two completely different systems, but I want you to notice something. Both pathways have the exact same goal. They're trying to answer one question. What protein should the immune system examine? The difference is simply where that protein came from. So I like to think this slide as the roadmap for the rest of today's lecture. Over the next few minutes, we're going to follow both of these pathways in detail. We'll start by looking inside the cell with MHC class 1. Then we'll move outside the cell with MHC class 2. And once you understand those two pathways, almost everything else in adaptive immunology begins to fit together. So let's begin with proteins that originate inside the cell, or the MHC class 1 pathway. Earlier, we learned that proteins produced inside a cell enter the endogenous pathway. Now let's follow that pathway step by step. Every second of every day, your cells are continuously recycling proteins. Some proteins have been damaged. Others have simply reached the end of their useful life. And if the cell is infected by a virus, viral proteins are mixed with that same recycling process. The structure responsible for this is called proteasome. Think of this as the cell's recycling center. Its job is to chop up proteins into smaller peptide fragments. And here's what's remarkable. The proteasome doesn't care whether a protein belongs to you or to a virus. It simply cuts proteins into pieces. Those peptide fragments are then transported into the endoplasmic reticulum through a protein called TAP. the transporter associated with antigen processing. You can think of TAP as a security checkpoint. Only peptide fragments of the appropriate size are allowed through. And once inside the endoplasmic reticulum, one peptide is loaded onto the groove of an MHC class 1 molecule. And that MHC peptide complex then travels through the Golgi apparatus and eventually reaches a cell surface. Now, every nucleated cell in your body is displaying a tiny molecular sample of what's happening inside the body. This is where CD8 positive cytotoxic T cells come into the story. As they circulate throughout the body, they inspect these peptide-MHC complexes. If they recognize normal self-peptides, they move on. Nothing is wrong. But if they encounter viral peptides or other abnormal peptides, they recognize that something inside the cell has changed. Rather than attacking the virus directly, the cytotoxic T cell eliminates the infected cell before the virus has a chance to spread. I like to think of the MHC class 1 as the cell's internal security report. Every nucleated cell is constantly telling the immune system, here's what's happening inside of me. So that's how the immune system monitors what's happening inside of our cells. But many dangerous microbes never enter our cells at all. How does the immune system monitor what's happening outside? And that's where MHC class 2 comes into the story. Now let's switch gears. Instead of looking inside the cell, let's look at what's happening outside the cell. Not every pathogen infects our cells. Many bacteria, fungi, parasites, they spend much of their lives in the spaces between our cells, circulating through our tissues and bloodstream. If the immune system only monitors what was happening inside of our cells, it would completely miss many of these threats. That's where the MHC class 2 pathway comes in. Unlike MHC class 1, which is found on almost every nucleated cell, MHC class 2 is found primarily on specialized groups of cells called professional antigen presenting cells. These cells include dendritic cells, macrophages, and B cells. Think of these cells as the immune system's investigators. They're constantly sampling their environment, looking for anything unusual. Imagine a dendritic cell encounters a bacterium. Rather than ignoring it, the dendritic cell engulfs it through phagocytosis, enclosing it inside a membrane-bound vesicle. And inside that vesicle, digestive enzymes begin breaking down the bacterium into much smaller peptide fragments. And at the same time, new MHC class 2 molecules are being assembled inside the endoplasmic reticulum. But here's something clever. Their peptide binding groove is temporarily blocked by a protein called the invariant chain. Think of it like a protective cover placed over an electrical outlet. It prevents the wrong peptides from binding too early. Only after the MHC class 2 molecule reaches a specialized compartment does that protective cover get removed. Now, the bacterial peptides can be loaded into the binding groove. The completed peptide MHC complex is then transported to the cell's surface. When a CD4 positive helper T cell encounters that complex, it doesn't immediately destroy the antigen presenting cell. Instead, it begins coordinating the immune response. Helper T cells activate B cells. They stimulate macrophages. They recruit other immune cells. They're less like soldiers and more like conductors directing an orchestra. So I like to summarize the two pathways like this. MHC class 1 answers the question, what's happening inside the cell? MHC class 2 answers the question, what's happening around the cell? Two different questions, two different pathways, one remarkably controlled immune system. So far, the rules have been pretty straightforward. Proteins from inside the cell enter their MHC class 1. Proteins from outside the cell enter MHC class 2. But as we all know, biology loves exceptions to the rule. And one thing I love about biology is that just when you think you figured out the rules, nature shows you an exception. And in immunology, this exception is incredibly important. So imagine a virus infects only a handful of cells within a tissue. A nearby dendritic cell isn't infected itself, but it engulfs pieces of those infected cells. According to everything we've learned so far, those proteins came from outside the dendritic cell, so logically they should enter the MHC class II pathway. But here's the problem. Viruses live inside cells. To eliminate virus-infected cells, the immune system needs to activate CD8, positive cytotoxic T cells, which recognize peptides presented on MHC class I. So how does the immune system solve this dilemma? It uses an elegant workaround called cross-presentation. Certain dendritic cells have the unique ability to take proteins they've captured from outside the cell and redirect them into the MHC class I pathway. Those proteins are transported into the cytoplasm, processed by the proteasome, and moved through TAP, and eventually loaded onto MHC class I molecules. In other words, the dendritic cell presents an extracellular antigen as though it had originated inside the cell. Why does it matter? Because it allows the immune system to activate naive cytotoxic T cells before they ever encountered an infected cell themselves. So think of cross-presentation as an intelligence briefing. The dendritic cell has gathered evidence from the battlefield and now is warning the immune system about an intracellular threat that requires a cytotoxic response. This remarkable process plays a critical role in antiviral immunity, cancer surveillance, and even many of today's most advanced vaccines. Sometimes breaking the rules is exactly what the immune system needs to do to protect us. So now we've seen how peptide fragments reach the cell surface, but what exactly is a T cell looking at when it encounters one of these molecular display cases? We spent the last several slides following proteins through the immune system. They've been broken down into peptides, loaded onto MHC molecules, transported to the cell surface. Now we've reached the moment everything has been leading toward, recognition. At first glance, it might seem like the T cell receptor simply binds to the peptide, but that's only part of the story. The T cell receptor actually recognizes two things at the same time. It recognizes the peptide and the MHC molecule presenting it. Think of it like reading a message displayed in a picture frame. The message matters, but so does the frame. The T cell expects to see a specific message presented in a specific way. If either one goes wrong, the interaction may never occur. So notice the illustration on this slide. The peptide sits inside the groove of the MHC molecule like a message resting in a display case. The T cell receptor reaches down and makes contact with both. Part of the receptor interacts with the MHC molecule itself. Another part examines the exposed amino acids on the peptide. It's reading the message and verifying who's presenting it. That detail turns out to be incredibly important. It helps explain why organ transplantation is so challenging. When a transplanted organ enters the body, the recipient's T cells just don't encounter unfamiliar peptides. They also encounter unfamiliar MHC molecules. Even if the peptide looks relatively ordinary, the unfamiliar MHC molecule may be enough to trigger a powerful immune response. That's one reason physicians carefully match HLA types between donors and recipients whenever possible. So here's the key takeaway. A T cell never asks only what peptide am I looking at, it also asks who is presenting it. That partnership between the peptide and the MHC molecule is one of the defining features of adaptive immunity. But here's another interesting question. Every virus and every bacterium contains thousands of potential peptides. So why does the immune system focus only on a small number of them? That's where our next concept, immunodominance, comes in. Let's do a little thought experiment. Imagine you've been infected with a virus. That virus contains dozens of proteins, and each of those proteins can be broken down into hundreds of different peptide fragments. So here's the question. If thousands of peptides are generated through an infection, why doesn't your immune system respond to all of them? The answer is surprisingly simple. It can't. Your immune system has limited time, limited energy, and limited resources. Just like every other biological system, it has to prioritize. That process of prioritization is called immunodominance. So take a look at the funnel on the slide. At the very top are the thousands of potential peptide fragments produced during protein degradation. But as those peptides move through the immune system, they begin to pass through a series of filters. Some peptides never bind effectively to an MHC molecule. Others bind, but not strongly enough to remain on the cell surface. Some are presented perfectly, but there isn't a T cell available that recognizes them. Others generate only a weak immune response. And by the end of this process, only a relatively small subset of peptides produces a disproportionately large T cell response. These are called immunodominant epitopes. I like to compare this to watching the highlights from a football season. No one shows every single play from every game. Instead, the highlights focus on the plays that had the biggest impact. Your immune system does something remarkably similar. It focuses its attention on the peptide fragments most likely to generate an effective response. But pathogens are evolving too. Viruses mutate. Sometimes those mutations occur within immunodominant epitopes. And when that happens, the immune system may have a much harder time recognizing that pathogen. That's one reason influenza viruses and many other viruses continue evolving year after year. They're constantly changing the molecular targets the immune system has learned to recognize. So immunodominance is a balancing act. It allows the immune system to focus its resources effectively while creating an evolutionary arms race between pathogens and the immune system itself. So now we know why only certain peptides dominate the immune response. But that raises another question. What makes one molecule a better antigen than another? Now that we've seen how the immune system chooses which peptides deserve its attention, let's ask one more fundamental question. Why are some molecules excellent antigens while others barely get noticed? There isn't just one answer. Several different characteristics determine how immunogenic a molecule will be. Let's begin with size. In general, larger molecules tend to make better antigens. Why? Because larger molecules contain more potential epitopes. Think of it this way. A short children's book contains only a handful of memorable passages. A thousand-page novel contains hundreds. The same principle applies to proteins. Larger proteins simply produce more opportunities for immune recognition. Next comes chemical complexity. Simply repeating molecules usually makes for poor protein antigens. Proteins, on the other hand, fold into incredibly complex three-dimensional structures. These folds create unique molecular landscapes filled with grooves, loops, pockets, and exposed surfaces that immune receptors can recognize. The third factor is foreignness. Generally speaking, the more different a molecule is from your own proteins, the easier it is for the immune system to recognize it as something unusual. But the final factor is perhaps the most fascinating: host genetics. Every one of us inherits a unique combination of MHC molecules, also known as human leukocyte antigens, or HLA molecules. Remember that MHC molecules determine which peptide fragments are displayed to T cells. If two people inherit different MHC molecules, they may present completely different peptides from the exact same virus. That means one person may generate a very strong immune response, while another emphasizes different epitopes and mounts a somewhat different response. The virus hasn't changed, the way it's presented has. This helps explains why people respond differently to infections, why vaccine responses vary, and why certain autoimmune diseases often occur more frequently in individuals carrying particular HLA types. So when we ask, what makes a good antigen? The answer isn't just the antigen itself, it's also the immune system encountering it. Recognition is always a partnership. So far, we focused on the antigen, but there's one final question we haven't answered. Why does the immune system sometimes respond aggressively and other times remain completely tolerant? Everything we've discussed today might seem like basic science: proteins, peptides, MHC molecules, T cells. But here's the exciting part. Every one of those concepts has direct clinical relevance. Let's begin with vaccines. If you've already watched our vaccine episode, Vaccines, How They Really Work, you know that vaccines train the immune system before a real infection ever occurs. Now, you know how they do it. Vaccines introduce carefully selected antigens or even individual epitopes to teach the immune system what to recognize in the future. Understanding antigen recognition allows scientists to design smarter vaccines that focus the immune response on the most protective targets. Now consider blood transfusions. Your ABO blood group is determined by carbohydrate antigens on the surface of your red blood cells. If someone receives incompatible blood, the immune system isn't reacting to the entire red blood cell, it's reacting to those specific antigens. The same principle explains organ transplantation. When a transplanted kidney or heart enters the body, the recipient's immune system examines the donor's MHC molecules and associated proteins. If those molecular structures appear foreign, the immune system may begin rejecting the transplanted organ. Finally, think about cancer. Many tumor cells produce altered proteins called neoantigens. These are molecular structures that weren't present when immune tolerance developed. Modern cancer immunotherapies work in part by helping the immune system recognize these neoantigens more effectively. Although vaccines, blood transfusions, organ transplantation, and cancer seem like completely different areas of medicine, they're all built upon the exact same question. Does this molecular structure belong here? Once you begin asking that question, you'll start seeing antigens everywhere in medicine. So let's finish where we began, with one simple question. What does the immune system actually see? At the beginning of today's episode, I asked you a simple question. If I asked you to point to an antigen, what would you point to? So I hope your answer has changed. When you look at a virus, you see a virus. When you look at a bacterium, you see a bacterium. When you look at a transplanted kidney, you see an organ. But your immune system experiences the world differently. B cells recognize specific molecular surfaces on intact proteins. T cells recognize peptide fragments presented on MHC molecules. Different mechanisms, same goal. To determine whether a molecular structure belongs in the body. Everything we've discussed today comes back to that single idea. Vaccines work because they introduce recognizable antigens before an infection occurs. Blood transfusion reactions occur because incompatible blood cells display unfamiliar antigens. Organ rejection begins because donor tissues express unfamiliar MHC molecules. Autoimmune diseases develop when the immune system mistakes self-antigens for foreign ones. Cancer immunotherapy succeeds by helping the immune system recognize specific tumor-specific antigens. Those may seem like completely different areas of medicine, but underneath them all is the same remarkable principle. The immune system doesn't recognize organisms. It recognizes molecular information. And once you understand that, you've taken one of the biggest steps towards immunology.